Hello, I'm Dr. Azaz from medicovisual.com, and in today's visual lecture we will talk about why is pH maintenance so important. So you see, in our blood the pH is maintained at a very narrow range of 7.35 to 7.45. So such a narrow range is maintained. Why it is so important? The reason is that almost every physiological process in our body it is affected by the pH changes, and even a slight change in pH has detrimental and drastic effects on the different biological processes going on in our body. So it's important to maintain the pH in our blood, and the effects are cascading. What I mean by this is that, for example, this is a process from A to B. Let's suppose it is directly disrupted by the pH change, then this would be an ongoing effect, right? So B would be going to C, C would be going to D, and so on, D to E, just like a butterfly effect. You might have read it, right? So this is, if the problem is here, whole this chain of event will be disturbed. So that is why pH maintenance is so important. By the way, I can't even think of an exception of a biological process that is not affected by pH changes. It is so important that almost every biochemical and biological process is affected either directly or indirectly or both by the changes in pH. So that is why it is important. Now in this lecture, I will not discuss each and every process that is affected by pH changes, because it is practically impossible to discuss that. As I said, every process is affected, so I can't tell you in this lecture every process, every biological process that is going on in your body. But in today's lecture we will understand some basic principles related with the basic principles that related with the pH changes, that how exactly the change in pH affects our body, and then along with that we will discuss few important processes, right? So let's start the discussion. Now let's start it from the very basic concept that, let's suppose we have a chemical here, a compound here, or it may be a part of a compound. Let's call it a substance A, right? So it is having an atom. It could be any atom, but typically it is oxygen, but it can be any atom. Not any atom, like it can be one of the many atoms, right? Not any atom. So this is let's say some atom, and it is slightly negatively charged. It is having a partial negative charge on it. And then we have another substance or a part of a compound, right? So let's call it B. It's just arbitrarily I am naming it so that you can understand it. And it is having an other atom, and it is different from this one. So there is another atom, and it is partially positively charged, right? So normally what should be happening, that these two should somehow interact with each other. These two should, in these A and B moieties or A and B substances, they should somehow interact with each other either chemically or at the intermolecular level, like between the molecules there should be some bond formed. For example, hydrogen bond should be formed. This should be happening normally. But what if, and this might be, typically this is hydrogen, or it is atom, hydrogen atom here, right? So what may be, and typically it is oxygen, right? So what may be happening here, that let's suppose if pH is low, so there would be lots of protons lying here and there in this environment. Of course, blood is surrounding all these structures. Let's suppose, of course, it is happening in our body, so it will be surrounded with blood, and pH is low, pH of blood is low. What I mean by this is that, if the pH is low, it means that we have more protons in our blood. So what will happen that this partial negatively charged moiety or substance, what it can do is that it can pick up this proton, and it will lose this charge, right? And now will it engage with this substance? No, it will not engage with this substance. It is already paired with a proton, so now it has no interest in pairing with this positively charged substance here. So this is like a couple. So this is one member of the couple, this is another member of the couple. Let's suppose this couple, he picks up a random girl and he starts liking this girl. So will it have any interest to talk and engage with his other partner, right? So that's how it is going on. So here you see the effect of decreased pH. Similarly, in case of if the pH is increased, the protons are less here, then what will happen? This hydrogen, it may be released from here. This hydrogen may be released as a proton, right? So now it will lose this partial positive charge, right? So now again, these two will not engage with each other. And then of course the cascade of event will be affected like this. Now these two may be separate compounds or they may be part of a single compound, different parts of a single compound. This may be a polypeptide chain. And different components, different parts of polypeptide chain may be there, and they may fail to interact with each other with the changes in pH. Okay, this might be confusing for some of you people, but let me simplify it for you. I took it from the very basic concept. I started teaching some basic principles. Now we will see some examples. So what is the structure of polypeptide? Polypeptide. What is the meaning of polypeptides? Polypeptide means that we have amino acids. Let's suppose this is an amino acid. Okay, let me draw it for you. NH2, then we have C, and then here we will be having some side chain. Let's call it R, and it is actually a convention to call it R. It could be one of the many different types of R chains or groups that may be possible. Let's not go into that. It depends upon the type of amino acid, right? So then we have another carbon here, and then with carbon we will be having oxygen. This carbon is called carbonyl carbon, and here we have oxygen. So this oxygen is electronegative, so there will be a partial negative charge here. If you don't understand what is the meaning of electronegativity and partial negative charges, you can do two things. Either you will have to go deep into chemistry, you can study the chemistry books, or we have a video on chymotrypsin mechanism, and in that video I briefly described some aspects of chemistry as well, and in that lecture I have also described the concept of electronegativity. You can watch that lecture as well. So anyhow, let's move forward. Let's suppose we have another amino acid. Let's copy this, and then okay, here we have this amino acid. Now what will happen that these two amino acids, they can form a bond with each other. So here initially it was having 2H, two hydrogen. One of the hydrogen will be lost. There was this hydrogen will be lost, and it will form a bond with this here. This is called peptide bond. And of course hydrogen is having a partial positive charge. This was carbonyl carbon, not this one. Okay, let me label it for you. This was carbonyl carbon, and this is carbonyl oxygen, and this one is this is not carbonyl, I'm sorry. This is amino nitrogen, and this is amino hydrogen. So in this way, long chains may be there. If I draw this structure, of course I do not want to waste your time and my time by drawing it completely, but let's draw a long chain, relatively long chain, and of course it is made up of several different small amino acid subunits. But the thing that is common is that they will be having these repeated peptide bonds. You see, we will be having oxygen here like this, and then we will have this carbonyl oxygen, and we have amino hydrogen, again carbonyl oxygen and amino hydrogen, carbonyl oxygen and amino hydrogen. Of course this will be having, yeah, here we can draw the hydrogen as well. So partial positive, partial not positive, partial negative, partial positive, negative, positive, negative, positive, and so on. So you see we have these charges here. Now if we draw a long chain, what can happen that these can interact with each other. You see, these can interact like this, they can interact like this, or if the chain folds like this, so somewhere here hydrogen may be there, somewhere here oxygen may be there, they may interact like this. Here oxygen and hydrogen, they may interact like this in a perpendicular way, right? So if the hydrogen bonding forms in the parallel fashion, parallel to this backbone, this polypeptide backbone, then what will happen that a structure like this will be formed. I will show you in 3D, don't worry, you might get confused here. And if we have perpendicular bonds, then we will have sheets like this that might be formed. So a proper structure will be formed like this. Again, don't worry, I will show you in 3D, it will become much more clear. Now the thing is that again here you see we have partial negatively charged oxygen. If pH is low, then it might pick up proton before it could form this bond, or it can lose the proton. If pH is high, it can lose the proton. So in this way, this structure will be lost. By the way, this is called alpha helix, and this is called beta-pleated sheets. So these structures might be disrupted simply because of change in pH. Now let me show you this thing more clearly in 3D so it becomes really clear for you. Okay, let me show you. So here we have, this is a polypeptide chain. So you see, so this one is carbonyl. Here you can see this is carbonyl oxygen, this is carbonyl, this is amino hydrogen. So if they form parallel bonds, you see, if they form parallel bonds, parallel hydrogen bonds, so these dotted lines are indicating the hydrogen bonds. So parallel hydrogen bonds are formed like this. So that's how the alpha helical structure is formed. Don't worry, this lecture is not about the structure of protein, but to make the concept clear, I created these 3D models and 3D animations for you. And if we talk about the beta-pleated sheet, this is the structure of beta-pleated sheet. Let me show you. So again, polypeptide chain that consists of different amino acid subunits, and if they perpendicularly form the bond like this, so you see they have this. So here you can see these hydrogen bonds indicated with the dotted lines. So you see, of course, if these bonds are lost or these bonds they do not form at all, naturally what will happen that the structure of these alpha helix and beta pleated sheets it is disrupted, right? And these are the basic structure, the secondary. This is the secondary structure of the protein, and if the structure of protein is lost, its function is also lost. It cannot function properly. So the structure is required, a proper three-dimensional structure is required for a protein or anything to function properly. So imagine you have a car and it is totally destroyed. Can you drive that car? Will it run? Of course not. Same is true for everything in the universe. It needs a proper 3D structure to function properly. And it is not just about alpha helix and beta sheets. By the plated, I mean the structure of these sheets is like this three-dimensionally. The structure is like this, slightly corrugated sort of structure. These are the beta-pleated sheets. But anyhow, we will not go into details of structure of protein. That is not our today's topic. But the point is that the changes in pH will disrupt and can disrupt the structure of proteins. And proteins are not just alpha helix and beta-pleated sheets, but alpha helix may further be, interacting with each other and forming specific bonds. That is called the tertiary structure. And then tertiary structure, further subunits of tertiary structure, they are interacting with each other to form the quaternary structure. We will not go into those details, but the point is that even those structures are disrupted by pH changes. So the pH change, even slight pH change, has the ability to disrupt the structure of proteins. But why I am teaching you this thing? Why it is so important to maintain the structure of protein? The reason being that proteins are one of the most, rather, these are the most important, not one of, these are the most important macromolecules of your body. Many important structural and functional components of your body, they are made up of proteins. So your bones, they have lots of proteins, your hair, they have protein, skin, they have protein, you name it, everything in your body has something to do with protein. And there are many chemical reactions going on all the time in your body, and to make those chemical reactions feasible, you need protein. Why do you need protein? You need enzymes actually. And what are enzymes? Yeah, enzymes are also proteins. So everything that is required in our body to function and to form some important structure, it is protein. And pH changes can disrupt the structure of protein. pH changes can disrupt the structure of enzymes because they are protein. But it is not just due to the structural changes that enzyme might fail to work. Actually, there could be chemical disruption within the enzyme. So you see that we have chymotrypsin enzyme, a classical example, chymotrypsin. It consists of a catalytic triad. We have discussed these details in another lecture that chymotrypsin consists of three important amino acid residues. In the center we have histidine 57, and it consists of this ring, right, imidazole ring like this, and here we have carbon and carbon like this is the structure. And then we have an other amino acid residue called serine at 195 position. It has O and H. And then we have aspartate at 102 position. It has carbon and oxygen, and here we have hydrogen. Now here the hydrogen bonds are formed. Now what will happen as this reaction starts? The histidine, it will pick up this proton from here. It will pick up this proton, and now this oxygen will become highly electronegative. and then this will attack the substrate and do the reaction. We will not go into those details, but the thing is that to start this reaction, the histidine has to pick up, it has to pick up this proton from here. But what if pH is low and it has already picked up this proton, it has already picked up this proton from the environment? So will it engage in any way with this proton, with this hydrogen of serine? The answer is no. This hydrogen will be sitting here. Now the substrate will come and it will do nothing. Right? I hope you can understand. Again, the basic principle is the one that I highlighted here. This is the basic principle. If you keep this one in your mind, you will be able to understand whole of this lecture very nicely. So I hope you got the point. So it can, pH changes can disrupt the structure of enzyme, it can disrupt the functionality of enzyme by disrupting the structure, as well as through chemical disruption, as I just discussed with you in a classical example of chymotrypsin. Again, we have a detailed lecture on chymotrypsin. If you want to understand it more clearly, then you should watch that lecture, right? Now, another thing, our blueprint in our body, the genetic blueprint, the DNA, DNA is a double helix structure. It consists of one chain like this, a helical chain like this, and along with that, we have another chain like this. So it's a double helical structure. Now, how this double helical structure is formed? If we enlarge this part only, we will see that we have this one chain here, and then we have this other chain here. Now we have the what we call as the residues, the nucleotides. So let's suppose we have gone in here, so, and let's suppose we have cytosine here. Oops, sorry. So we have cytosine here, then we will have then cytosine here, and we will have gone in here, and they will be having the hydrogen bonding, they will be having hydrogen bond between them like this. And again, the hydrogen bond, how it is formed. Here there might be, let's say, oxygen, and here there might be hydrogen, as we have discussed earlier, and they may form this intermolecular bond with each other. Again, this bond can be disrupted, as you might logically assume by now that this bond might be disrupted, or it may fail to form with a drastic change in the pH. Now, fortunately, with slight changes in pH, your DNA is not going to get disrupted. You need a very, very high changes in pH, like pH less than 3 or maybe higher than 11. At that much, pH changes, the structure of your DNA might get disrupted. But you like it or not, unfortunately, you might even die before that happen. Right? So in some textbooks, while discussing the importance of pH changes, they do describe the effect on DNA helix, but this does not happen at in routine. Like if you might develop somehow, God forbid, you develop the alkalosis or acidosis, it is not going to damage your DNA, fortunately, at this level, at this much level that it becomes uncoiled like this, right? So but it is mentioned in textbook, so that is why I described it. But as a general principle, you must understand it. Secondly, the more important thing that might happen in your body, if there is drastic changes in pH, not very drastic changes in pH, rather I should say like that, then we have other enzymes, the polymerase enzymes, we have helicase, topoisomerase, and different enzymes that are required to copy the DNA. There is transcription factor, different things that are required to manage and for the functioning of your genes. Those might be affected by pH changes. Not this unwinding thing, but those might be directly affected by pH changes, as I have discussed previously, by the virtue of These enzymes being protein, their structure might get disrupted, right? So yeah, I hope you are clear about this. Now another thing that I would like to discuss, that pH changes also affect membrane potential and resting membrane potential as well. What I mean by this, let me tell you. As Dr. Najib has famously said that our cells, they are the bags of potassium. Our cells, they are bags of potassium. So we have lots of potassium filled inside our cells. These are the bags of potassium, and these bags of potassium, they are surrounded by salty water, sodium, high in sodium. So outside extracellularly we have lots of sodium. As they say, life originated in the deep sea. So cells became potassium bags, and outside that we had that sea water, right? So now what happens is that normally we have some channels, some pores here, and these are called potassium leak channels. So we have potassium leak channels. So what will happen then? Outside, outside the cell, we have very little potassium. Outside the cell, we have very little potassium. Inside the cell, we have actually lots of potassium. So as these channel appeared, what will happen, that potassium will rush out of the cell. Potassium will rush out of the cell. So we have loss of this positive charge, positively charged ions inside the cell, they are lost. So what will happen, that inside the cell we will have a negative charge that will develop. Of course, not all the potassium will come out, or there will not be a balance of potassium inside and outside, because due to presence of this negative charge, this electrical gradient is established, and this negative charge will prevent more potassium from going out. Again, these things are the detail will be discussed in details of physiology, membrane physiology. It's not the today's topic, but the thing is that normally at rest, the inside of your cell, it is more negative than outside. So it is different for different cells, but generally it is nearly about at -70 millivolt. This is almost here, -70. Now what will happen, that let's suppose this cell is a touch receptor, or heat or cold receptor, but any receptor it could be. So let's suppose we touch this cell here, not directly, but somehow we try to stimulate this cell here. We rub this cell from here. Then what will happen, that normally this cell was not significantly permeable to sodium. So some sodium, with touching or with some disruption, some sodium might leak inside. So what will happen, that from -70, what will happen, that the charge will go from inside, the charge will go slightly up, -20, -30, 40, -20, somewhere, right? So at certain level, let's say -20, I don't know the exact number, let's say -20, another type of channel opens, and these are called sodium channels. Sodium channels momentarily open, and they open at specific voltage. They open very quickly and then close, and when these channels open, the sodium will rush in, and then we will have a formation of sudden spike in potential, spike in charge, and then these channels will close and some other channels will open, then this will be repolarization. So this is the action potential. Normally -70, then due to some leaking of sodium or other positively charged ion inside the cell, the charge will slightly go up and up at certain degree. This is the threshold of opening of these fast fast voltage gated, because they are affected by voltage. Fast voltage gated sodium channels, sodium channels, there will be sudden spike in charge towards positive side. Let's suppose positive, I don't know, 100 or whatever. So it will move towards positive and cell will be depolarized. Initially it was polarized, inside was negative, now it inside will become positive, so cell will be depolarized. I hope you got the point. Why I am teaching you this detail, let me this will become very clear now. Let me tell you. Now, imagine you develop a condition called alkalemia or alkalosis. Alkalemia, which means that you have increased pH of your blood, which means that protons in your hydrogen ions or protons in your blood are less, decreased in proton. So to compensate this, what your kidney will do is that this is a nephron, a single nephron, just for the sake of representation. What it will do is that it will it will try to capture the proton, it will try to bring the proton inside, right? So positively charged ions, it will try to bring it inside and to prevent these protons from going into urine, right? So protons will be brought in, and to maintain the electrical neutrality, some other positively charged ion should go out, and as it turns out, the other positively charged ion that goes out and lost in urine is potassium. So this is potassium hydrogen exchanger. So it will pick up, bring in the hydrogen, and of course it will come, hydrogen will come into the proton will come into your blood. So it will bring the proton to compensate this decreased hydrogen or proton, and it will push out the potassium inside the cell, inside, not inside the cell, inside the lumen, sorry. And similarly, many of the cells of your body, they will do the same. They will activate these channels, the exchanges, that hydrogen will go out in blood and potassium will move in. So ultimately what it can do is that it can cause decreased potassium in our blood. In our blood, it can lead to what we call as hypokalemia. Hypokalemia is decreased potassium in our blood. So what will be the effect of hypokalemia? It means that the potassium here will be less outside the cell, potassium here will be less, and inside the cell potassium here will be high. So what it will do is that initially, if less potassium was to come out due to this increase in concentration gradient, more potassium will move out, might move out through these leak channels. And then if charge was, let's say, initially -70, then this charge might be at let's say -90. So it would be like somewhat here. Now, will this be easy to stimulate this cell, that if there is even slight leaking of sodium, will it cause the opening of voltage gated sodium channel? No. This cell is hyperpolarized. This is hyperpolarized. It needs much more leaking of sodium or other positively charged ion inside it to activate the sodium channel and cause the action potential. So we were talking about alkalemia or alkalosis. So in alkalosis, there might be hypokalemia, and hypokalemia might lead to hyperpolarization of the cell. That resting membrane potential is more negative, and it can lead to difficulty activating the cells, especially the neurons, difficulty in action potential, right? Wrong. What if it if I tell you this is wrong? This is the biggest plot twist in our lecture. This is not true. Unfortunately, the things are not that simple. What actually happens that the nerves become more easy to activate in alkalemia or alkalosis. So much so that there might be sudden uncontrolled muscle twitching, right? This is called tetany. So tetany might be there. So did I teach you everything wrong? Well, no. Actually, this mechanism is there in its full glory, but we have another mechanism as well that is going on. So as I've told you about these channels, these voltage gated sodium channel, we have something that keep these channels in check. We have calcium here. So calcium bind to these channels, and calcium acts as sort of a brake for these channels. Calcium try to control these channels, and calcium try to prevent excessive and aberrant activation of these voltage gated sodium channel. Please remember this thing: calcium prevents aberrant, I mean abnormal activation of these voltage gated sodium channels, and calcium stabilize these channels in closed state. So calcium is sort of physiological brakes for these channels, and if calcium is low, this brakes might be removed, and then these may become aberrantly activated, abnormally activated. So lots of sodium influx will be there, and then this may lead to abnormal and excessive action potential generation in our nerves, in our muscles as well, which lead to muscle twitching, which might lead to epilepsy like situation. That is, and this situation, muscle twitching and stuff like that, it is called tetany. So, what the calcium has to do with alkalosis? Well, actually, there is something interesting going on, that we have an important plasma protein, the most common plasma protein in our blood, that is albumin. So, this is our beautiful beloved albumin, most important plasma protein. So this plasma protein has different amino acids, and within these amino acids we have protons attached with them. I think I will use another color. So you see, you might have seen the apple tree. So these are like apples growing on apple tree, right? So we have lots of protons attached on these albumin, on the albumin molecule. So if pH is low, and in alkalosis pH is low, of course, not low, pH is high, I'm sorry about that. So pH is high and proton is low, concentration of proton is low, so albumin might act as a hero. It says that, okay, we have less proton in our blood, why not I give my protons to the blood? So it will release the proton in the blood. It is part of blood anyway. It's a plasma protein flowing in the blood, in the plasma. Plasma is, by the way, the liquid part of our blood. So it will release some of the protons in the blood, right? So as it loses the proton, the space, these spaces are there, these are left vacant, and there is loss of electrical neutrality, so they pick up another positively charged ion, and that positively charged ion is calcium. As it turns out, normally the albumin is not just carrying the proton, but albumin is bound to lots of calcium as well. It is normally what happens, that albumin is normally bound to calcium. But in alkalosis, as more and more protons are lost from the albumin, right? More and more protons are lost from the albumin, this albumin molecule will pick more and more calcium from the blood. So it means that within the blood, the amount of free ionized calcium will become low. So it might lead to hypocalcemia. And hypocalcemia can do what? It can it will, if there is decreased calcium, these brakes will not be working. So then this will lead to aberrant and abnormal activation of voltage gated sodium channels. So two mechanisms are going on in your body in case of alkalosis, and exact opposite is going on in case of acidosis. So what will happen? One process is that alkalosis lead to hypokalemia, which leads to hyperpolarization of neuron, and this cause decreased activation of neuron. On the other side, we have the same alkalosis, and this leads to albumin and stuff that I've just discussed. This lead to hypocalcemia, and brakes are lost, and this lead to aberrant activation of the neutrons. No, neutrons. What I am saying, I'm sorry, neurons, not neutrons. So aberrant activation of neurons. Now this mechanism of hypocalcemia, it overpowers the other mechanism. So that is why normally this is more, more potent mechanism that is going on. So net result is that there is increased and aberrant activation of neurons, leading to excessive stimulation of neuron and excessive development of action potential in alkalosis, right? And in acidosis, the stuff is reversed. I hope you can understand this thing. So both mechanisms are going on. The tug of war is going on between these two mechanisms, but this mechanism generally wins. And it's a hypothetical idea, or you can say a thought experiment, that because this mechanism exists, it is very likely that the tetany and other situation that might be going on in alkalosis, it may not be that much marked, may not be that much severe due to presence of this other mechanism. So this other mechanism might be keeping this process in check, right? So these are few things that go around in when there are changes in pH. Of course, there are many other things that might be going on. This lecture can go on and on for hours if I continue to discuss, but I hope you have a greater intuition of these things than as compared to what you had before watching this lecture. So that was all for today. Thank you so much for watching this video. Have a great day. Goodbye. And finally, yeah, I forgot to mention that, as I've told you, everything is affected by changes in pH. Even our organelles, cellular organelles, they are also affected. The cytoskeleton is affected. The mitochondria is affected. I think I would not want to go into their details, but it's everything, literally everything is affected by changes in pH, and that is why pH is maintained in a narrow range. So later we will discuss that how exactly the pH is maintained in such a narrow range. Thank you so much for watching this video. Have a great day. Goodbye.